Now, we are going to discuss issues regarding to the immune system and how the immune system interact with the pathogen and what happens thereafter to allow the pathogen to survive. This is an important lecture, especially for those people who are going to become doctors. We always wonder how come the pathogen is evading the immune system. Now, there are a number of terminologies which we need to understand. One of them, and you've been taught in microbiology, is what is a pathogen. And any organism which causes a disease, that becomes a pathogen. And this, as you've been taught in microbiology, could be the bacteria, the viruses, and other parasites. Bacteria and viruses are taught in microbiology, and other parasites are taught in parasitology, the so-called Department of Microbiology and Parasitology. Now, our duty in immunology is, once you've understood about all the pathogen which causes disease, is for us to tell you how these pathogens do interact with immune systems. And we have taught you previously that the first encounter any pathogen does with the immune system is what we call innate immune system. And if the innate immune system is not able to manage the pathogen, then the adaptive immune system will kickstart and will mount either the antibodies or the T killer cells. Now, between the innate system and the antibodies and other immune system killer cells and all that, sometimes they are not able to actually handle them, the pathogen. And when the pathogen is not handled, then what you get is what we call the disease, merely the survival of the parasite. If the immune system is able to manage the pathogen, to control the pathogen, then you do not get the disease. Now, and therefore, there are two additional terminologies which you need to understand: the susceptible host and a host who is not susceptible. And susceptible host is the one whose immune system will allow the parasite to survive, meaning that the parasite is either adapting or has had measures which counteracts the immune system. Now, in a host where the parasite is killed, more often than not, these will be eliminated through the innate immunity. And for the parasite to survive, many of the parasites have adapted different strategies to survive. Remember, we have lived with multiple pathogens for a long time. And with the time, they have learned to live with the immune system and how to avoid the immune system. Now, so this is essentially what happens when you get an infection, that the pathogen will try to gain entry to the host. The immune system will be activated. The immune system may actually clear the parasite or not clear the parasite. If it doesn't clear the parasite, then you get an infection. If it clears the parasite, then you do not get an infection. And at the end of the day, you see nothing. But if you get an infection, what you see is what we teach you, what is called the disease, the immunopathology, the disease. So it's important to think about all that when you are under study. Now, how this evolves depends on various factors. One of them is age. Indeed, we know very young people and very old people are very susceptible to some of the pathogens, especially bacteria and viruses. And this is because very young people, their immune system is not well developed, and they are not able to cope up with multiple pathogens which may infect them. Now, when you become very old, your thymus, where the T cells are actually trained, are not very good, and therefore elderly people tend to suffer from multiple infections. We know about gender, especially differences in infections between females and males, differences which may be because of the hormone profiles. For example, if you take a lot of antibiotics, we know some people will suffer from candida. We know if you are actually during different phases of female, they can be predisposed to candida infections merely because of actually gender. Now, there are the physical conditions which predispose you or make you more susceptible to infections. We know people who are malnourished, people who are not feeding very well, they are likely to suffer from multiple bacteria and parasitic infections. We know if you do suffer from extreme fatigue, sometimes you can be predisposed to certain pathogens. And if you are exposed to certain elements, heavy elements, they may actually affect the immune system, and you end up suffering from some infections. And also when your normal body functions are weakened, your resistance will be affected. We know based on your immunity, which you can gain either after an infection or after immunizations, the pathogens which you have come across before or after immunizations may not be able to infect you. And obviously, we know about genetics, that certain diseases are more do occur with the people carrying certain HLA. And we also know that if you have certain HLA, you may either become resistant to disease, or if you're infected, you may end up getting severe infections. A good example, which now is common knowledge, is HIV. We know there are people who are resistant to HIV, and you know there are people who actually progresses very quickly, and others progresses very slow based on the HLA genes they carry. So these are important factors which you need to consider when you're talking about immune evasion mechanisms. They are important for you to always remember that they are actually there. Now, all those pathogens, be it bacteria, viral, and other parasites, they use different mechanisms. And I'll try and run through these based on the types of the parasite pathogens you may come across. And the reason I'm doing this is more often than not, when people ask you questions, they want to ask you, how do the bacteria evade the immune system? How do the viruses evade the immune system? So I'll make it easy for you so that you can be able, if you are asked that question, answer directly without a lot of struggle. So let's start with what we call bacteria. You know from your microbiology that there are bacteria which are extracellular, and there are bacteria whose habitat is more often than not intracellular. But please do not forget that all of these, including viruses, they will move to the time of actually recognition, attachment, and then entry to the host. Those who actually live outside the host cell are the ones which we call extracellular. But one time or the other, there will be attachment. Those who live within the intracellular compartment of the host are actually intracellular. And these will be mostly actually a few bacteria and viruses. Now, because of that, the extracellular, the pathogens which are extracellular, they have to learn to evolve strategies which will actually, first of all, ensure they are not recognized by the cells. Now, the cells recognize the pathogens based on pattern recognition receptors. Those ones you've been taught. So the extracellular bacteria have to learn or avoid being detected by the cells which have pattern recognition receptors. This second strategy they have to evolve is many of the extracellular bacteria are eliminated either through the antibody, the complement systems, and other peptides, which may actually be produced, things like lysosomes and all that kind of stuff. Now, so it is important if they are going to survive that they avoid being destroyed by the antibodies. They avoid being destroyed by the complement systems. Now, so those are some of the things which you need to understand. When it comes to intracellular bacteria, remember the phagocytosis, the phagocytes. They are living within the host. And there are all those different strategies which the host is going to utilize to destroy the intracellular bacteria, starting with the invagination, fusion of lysosome to fusion, making the phagolysosome, the release of enzymes, lytic enzymes, which destroys. So an intracellular bacteria which has is clever will avoid or will actually make mechanisms which will make the fusion of lysosome and phagolysosome difficult, or release of the enzymes which are going to be released so that it doesn't have to be destroyed, or it will stay in places where any of those will not be able to. So let's see what happens with all that. Now, this you may not see, and I've just actually very well, but I think you can. I've just tabulated a number of mechanisms extracellular bacteria are going to use. One of them is purely shedding or inhibiting the areas which enable them to be recognized. One of them could be having capsular polysaccharides, and you know the bacteria which have capsular polysaccharides. This makes it very, very difficult for lead to be destroyed. So detection of LPS will protect, blocking the detection of LPS will protect the bacteria from the immune system. The bacteria which is very well known for that is pneumonia. Now, you can actually have hypoacylation of lipid, and Digvalis is one of them. And if you look at here, the lipid is preventing the toll-like receptors, which are pattern of recognition from actually it is blocking that. Now, the second one is you can coat the bacteria with self-proteins. You can coat the bacteria with self-proteins. So you could go and pick a protein which knows the host does express. Fibrin is one of them. It's used for healing, making the clot. The bacteria can cover themselves with that fibrin. And therefore, it means any cell will not be able to recognize that bacteria because it will be seen as self. Now, the second strategy which they use is what we call antigenic variation. Antigenic variation. It simply means, and you come across this also within the viruses, it simply means you are shifting what you are expressing today. Tomorrow, you express different molecules. And therefore, by the time you make antibodies against the prevailing variant, you are actually those antibodies are a bit weak. Or you can block the antibodies so that they become ineffective. Neisseria gonorrhea is actually very common, commonly uses that strategy. The third strategy is inhibition of opsonization. You inhibit opsonins. Now, you could do this simply by secreting complement degrading factors such that the complements are not able to work. Or sometimes you could actually have a capsular polysaccharide which blocks fixations. So a number of bacteria which are capsulated have that mechanisms. The other one is you can actually express Fc binding surface molecules, which means the effector portion of the antibody is not going to work. And these effector portions, since they are going to be found on immune cells, especially phagocytes, if they are not working, it means phagocytosis is not going to occur. And therefore, that pathogen may actually survive. There are issues of for the strategy of inhibition of reactive oxygens. You know, oxygens are very important. Reduced oxygen ions are very important in eliminating certain pathogens. Now, the way you do this, you can actually have secretion of an enzymes which degrades the oxide molecules which are formed, So S. aureus is very good in this kind of maneuver. And the end result is the enzymes, the superoxides will not work. And therefore, degradation, which will likely occur by having reduced ions, will not be able to destroy the pathogen. And obviously, there's resistance to peptides. You can modulate the cell membranes. And once you modulate the cell membranes, then you prevent the binding, and therefore actually meaning that the functions of AMP, adenosine monophosphates, is not going actually to work within that. So the energy will not be able to be provided. So those are for just to remind you, shedding or inhibition of MAPs, antigenic variation, inhibiting the opsonization, inhibition of reduced oxygen molecules, and resistant to antimicrobial peptides. Now, these are actually more often than not used by extracellular bacteria. And they are worth going to explore further as you sit and revise in different places. So which means you moving through this arm and going all the way to microbiology to know which bacteria will utilize each of these. So we are just guiding you on the areas which you need to concentrate on. If you go to intracellular bacteria, they also have various mechanisms. Remember, these are living within the cell. They are living within the cell. So the mechanisms which they have evolved are geared to enabling them survive the hostile environment within the host cell. And you have to work out what hostile environment we are talking about. You could do this merely by following the process of phagocytosis and what happens, following what is in any of the compartment, which each of the intracellular bacteria will likely occupy, following the type of the cells, which will likely be invaded by some of these intracellular bacteria. Macrophage is a good example for you to start because a number of these bacteria will actually try and affect the macrophage. So there are a number of strategies which they use, and I've tabulated, and these are not the only ones. I've tabulated 5 of them, 6 of them. Antigenic variation, we've seen it in extracellular bacteria. The inhibition of MAPs, we've seen it also actually within the extracellular bacteria. So those two can be used by both intracellular and extracellular. So you just require to know how they modulate the pili or fibrilli. So you need to know the bacteria which have a pili or fibrilli, and Salmonella is one of them. So, and when you modulate the area which allows the immune system to produce effective antibodies, then it means the antibodies which are actually going to be produced will not have any effect into the pathogen. So that antigenic variation, by the modulation of the areas, the areas which allow the effective antibodies to be produced, will render actually that survival of that bacteria. Now, the production of peptidoglycan hydrolysis is well known, as well as secretion of intracellular toxins by various bacteria are very well known. And therefore, the reason they survive within the host is merely because they can produce peptidoglycan hydrolysis, which can actually cause hydrolysis of some of the mechanisms which are important. Now, the issues of modulation of cell membrane has also actually been eliminated in the other extracellular bacteria. So what I want to concentrate more is issues which you understand very well, and I alluded to this. One of them, if you are living in a host bacteria, is you need to understand a host cell. You need to understand what mechanisms the host will use to destroy you. So any bacteria which lives intracellular knows very well the phagosome has to fuse with the lysosome. And when that fusion occurs, it will form phagolysosome. And the latex enzymes which are within phagolysosome are going to be dangerous for any bacteria which lives within the host, which will allow that. So obviously, a smart bacteria will be to try and have mechanisms which can inhibit fusion of phagosome and lysosome. Because we know very well it is that fusion which will kickstart the second mechanism of release of latex enzymes. So the bacteria which do that is tuberculosis, lepri, and pneumonia. They are very good with inhibition of fusion of phagosome and lysosome. And therefore, merely because that fusion is not going to occur, then their survival is guaranteed. Now, if that fusion occurs, then you need to have a mechanism to protect yourself from the latex enzymes which are going to be released. And one of them is just cover yourself with a chemical which can prevent the latex enzymes. So you could actually cover yourself with wax chemicals, hydrophobic cell wall, containing mycoric acid and other lipids. And this actually allows the bacteria like tuberculosis and lepri to be protected against the enzymes. The third mechanisms which some bacteria have used is, yes, the fusion has occurred and the phagolysosome has been formed. Yes, there are actually latex enzymes which are going to be released. Can I quickly escape from that environment? And yes, there are bacteria which escape from phagolysosome. And Shigella is one of them. Shigella is one of them. So you escape from the hostile environment. Or you could produce a chemical which enables you to destroy the entire hostile environment. So, like if you produce chemicals which can cause lysis of phagolysosome, production of hemolysins is one of them. That enables the Shigella to continue surviving within and therefore the functions of a phagolysosome will not be effective. So what I'm alluding to is for you to be able to work out the mechanisms the intracellular bacteria use, you need to go back to the processes of phagocytosis. And you'll be able to work out each and every step which can be prevented so that the bacteria can survive. That is how you work out that. Let me talk now. Those are bacteria, and I'm just highlighting key things. You may go and we may ask you about viral immune evasion mechanisms, very common. Now, you need to understand that viruses are obligate pathogens, very small pathogens. And many of them only survive within the host cells. And you need to understand they are divided into DNA and RNA bacteria. So you can have a DNA virus and RNA virus. Now, each of these will try and have different mechanisms which they are going to use so that they can survive. And one of them is purely, actually, yes, I have done infection. I'm going to go to sleep so that I'm not be able to evoke anything that I'm surviving. We call that latency. We call that latency. So the virus is not actively replicating. And because it's not actively replicating, the immune system is not able to handle it. You need to understand that several viruses do have latency. Some of them, the ones you need to know, herpes simplex virus, Epstein-Barr virus, varicella zoster, and CMV. They are very good in actually having latency. And during the latency stage, they are not causing any damage. They are not replicating, and therefore the immune system will actually not be able to be active against them. When they lose that latency, then they start replicating, and that's when you know you have herpes or CMV. That's when you know. And the immune system starts working. Now it will work against all that and mount a strong immune mechanisms, but some of the viruses will still actually survive and go into latency. And that is why some of these viruses are difficult to eliminate. Now, that's one very important, latency. It allows these viruses to evade the immune system. Now, the second one is inhibition of humoral immunity. You know very well that the antibodies are good in neutralizations and complement system activation. So some viruses will have encoded the FC receptor. They have encoded FC receptor, which will trick the FC portion, the antibody FC portion to compete with it. And therefore, the FC portion of the antibody will not be able to function. Others will encode complement receptor, and others will have control proteins. All this is with the aim of cheating the immune system. You cheat the immune system so that the real factor which is supposed to work does not work. Now, so that is important. And therefore, you may produce antibodies which are not going to work because it has been cheated. The cells are binding to the false FC receptor or the complement receptor, and therefore that becomes the critical. The third mechanism, which is important, and we see that with the influenza, is what we call antigenic variation. And within influenza, and I'm hoping that the people of virology have taught you this, you can have antigenic drift, which is a point mutations of the genes which codes the service glycoproteins. So you are permanently mutating. And therefore, when you are mutating, it means that the immune system cannot actually catch up with the mutations which are occurring. And every time the antibodies are produced, the virus actually have shifted to a different form, which we call antigenic drift. Now, the other one which can occur is what we call antigenic shift, and purely is reassortment of some parts of the RNA, so that you can reassort your genetic material and change it to a different one. Maybe two different variants can combine to form the genetic material, which means technically that again the immune system will not be able to recognize the new form of reassortment which has been formed by reassortment of the RNA genome. So antigenic variation is very important. It is affecting, it will affect both individual and an individual who is infected with a virus which has undergone antigenic drift or antigenic shift will end up infecting other individuals, and therefore it end up affecting the population. So mechanism number 4 is simply producing inhibiting cytokines. We know cytokines are important in recruitment and activation of immune cells. We know cytokines do bind to receptors within the cells, and when binding to the receptors, that's when actually their effect is felt. If they don't bind to the receptors, then they are quickly destroyed by our body. So you could simply actually produce receptors, force receptors, which are virally produced force receptors, which the cytokines will bind to. So they will not bind to the real cells, they will bind to the force receptors, which means recruitment and activation of those cells will not occur. These receptors, force receptors, may be chemokine receptors, may be interleukin-1 receptors, tumor necrosis receptors, interferon gamma receptors. And when that occurs, it means the chemotactic mechanisms is inhibited. So cells will not be able to move and roll over to where the virus infection is found. If it is interleukin-1 receptors, it means actually that the expansion of the immune cells will not occur. If it is tumor necrosis factor and interferon gamma receptor, you know they have antiviral effect. It means their effect is actually is not going to be effective. And if it is receptors which command the cells to move over and cause addition to the cells for the entry, it means they are inhibited. So the entire system is meant to cheat the immune system that something is happening, but it's a force. And therefore, immune system will sit there and nothing will occur. The other thing is what you call transcription factor decoys. you know very well that actually the virus enter the body, and many of them will use host factors for them to make new viruses. So if you want to eliminate the viruses, the immune system makes proteins through what you call transcription factors, including enzymes, including some of the things we are talking about here, chemokines. So if you can have decoys produced by the viruses, it means you are counteracting the normal mechanisms which produce factors which can actually eliminate the viruses. And number 5 is simply actually interfering with antigen processing and presentation. You know that the intracellular pathogens are processed, picked by MSC genes, and expressed on top of the cells bound to MSC genes. It is only then the T cells, cytotoxic T lymphocytes, CD8 T cells, will come and recognize the peptides which are bound to the MSC genes. So if you can inhibit the processing of the peptide MSC complex, it means the peptides are not going to be expressed on the cell surface. And therefore, the immune system, cytotoxic T cells, are not going to work on the cells which are having an intracellular infection, simply because there is no evidence that there is an intracellular infection. This second one is to inhibit proteins which transporter-associated proteins, which help in transporting different proteins to the cell surface. You inhibit that, there is no evidence there is an infection. Number 7 is simply actually many viruses cause inflammation, and inflammation is good as an immunological process. First of all, to ensure that the infection does not spread from the foci of infection to the distance. So if you can, some viruses do inhibit that process, enabling them to quickly spread to other areas because they have produced chemicals which inhibit inflammatory response. And number 8 is simply cause immunosuppression. And you can cause immunosuppression by different mechanisms. One of them is infecting the cells which fight the pathogens, things like CD4. And we know the famous disease which infects the virus which infects CD4 T cells, HIV is one of them. Or you can actually cause immunosuppression by producing virally encoded cytokines, which can cause immunosuppression. And therefore, the host is not able to manage the virus. So these are important things which we all need to understand how they work so that we can be able to handle certain parameters. Now, I did mention the parasites because the topics is general, and I'm going to try and use a number of them. And parasites, size is important. There are parasites which are very small and others are large. So they are large, and all these are taught within the parasitology. So protozoans, things like Histolytica, Plasmodium, Leishmania are important for you to know. So how do they actually evade the immune system? One of them is what we call anatomical secretion. Anatomical secretion. So purely a good example, live intracellular. When you're intracellular, you avoid the immune response. So like Plasmodium, we live inside red blood cells. And we know very, very well that red blood cells are not going to be recognized by T cells because there is no HLA antigen expression or natural killer cells. Others will live inside the liver cells, other stages. And therefore, it's important for you to follow the pathway of what happens every time Plasmodium does infect a host. Things like Leishmania and Trypanosoma cruzi will live within the macrophage and therefore avoiding the destruction by the immune system. So anatomical secretion is important. Then there is, and one of them have just repeated, the other one is antigenic variation. The different stages of Plasmodium will evoke different antigens. Which means every stage of the Plasmodium is telling the immune system produce these mechanisms. And then by the time you produce maybe an antibody to cope up with the extracellular Plasmodium, you will find it has actually gone to a different life cycle or inhibited or affected a different cell. And therefore, it is always one stage ahead. It's always one stage ahead. And therefore, the immune system is not able to eliminate the second stage, which has actually moved to. That's how Plasmodium manages to move from red blood cells to liver stage cells and all that kind of stuff. You could do the shedding of your service proteins, or you replace them so that every time you shed, the immune system is supposed to evoke a different mechanisms, and you are actually running the immune system every other time by different stages. And Tamwemba Histolytica is important in that aspect. There are others which cause the immune suppression. You manipulate the host immune response, and Plasmodium again will do that. And the others which will have different anti-immune mechanisms, things like antioxidase production, which prevents macrophage oxidative burst mechanisms, so that Leishmania will continue to survive. Now, there is one which is very interesting, which all of us requires to understand. These days, you rarely hear about the sleeping sickness because I don't know what happened to trypanosomes. The African trypanosomes has what we call service glycoproteins. Now, the service glycoproteins, there are over 2,000 genes which are involved in production of the service glycoproteins. And each of these genes can be able to produce a different service glycoproteins. Our immune system recognizes actually the service glycoproteins. It is what the immune system evokes. But these genes are permanently producing different service glycoproteins. And therefore, because of the changes in VCG, because of the changes, it allows the parasite to survive. It allows the parasite to survive. You exhaust the immune system because of merely the number of genes which are involved in producing the service glycoproteins. When the immune system is handling one service glycoproteins, the antibody being produced for that, a second gene kickstarts to produce a different glycoproteins. When the antibodies are covering that, another one comes. And at the end of the day, this is what you end up having. So you end up having a service glycoproteins is being produced, the antibody handles that, then a new one is being produced, and so forth. Antibody handles that, and a new one, and so this is a cycle. It's a cycle. It's a cycle. This was worked way back in 1910. That's why I put this. So it's very difficult for the immune system to eliminate the trypanosomes within the host. And at the end of the day, the host will die, the person will die because we are not able to eliminate that. So it's an important one to understand. The large parasites, helminth is one of them, also have evolved certain mechanisms. They have, and I'm running very quickly. One of them is merely the large size, which is difficult to eliminate, large size. The second one is purely coating with the host proteins. You coat with the host proteins like RBCs, which means that the immune system will not recognize the helminth as actually a foreign pathogen. The third one is what we call molecular mimicry, that the structures of the parasites will mimic those of our host, of our body. And therefore, that allows the evasion of the pathogen. Number 4 is anatomic separation, whereby actually larval stage will live in certain places inside the mammalian muscle. And the fifth one is shending or replacement. We've seen that you shend part of your service antigens, so new ones are formed. And the 6th one is immunosuppression. We discussed about that. And number 7 is anti-immune mechanisms, like secreting the enzymes that will cleave the antibodies which are produced, and therefore ensuring that antibody does not actually does not do its functions. And number 8 is simply migration from one place to the other, from gut, to avoid local inflammations. And number 9 is production of certain parasites, the enzymes, which will make it resistant to ADCC and oxidative stress. These 9 are important for you, and I'll send these slides for you to understand them so that you can be able to work out through certain strategies. Now, it's 9, and I think I've run through the helminth very quickly, but I want to ask if there is a question before we start running late for other activities. Are there questions? Class lab?
Yes.
Are there questions?
seems that there are no questions on the chat box, but if anyone has a question, they can just unmute themselves and ask.
Okay, very good. Then I'll send this presentation to you. It's very important, especially because of viral helminth. If you hear what I'm talking about, eh?
Yes, yes.
Yeah.
Okay, thank you very much.
Okay, you're welcome. Good. I don't see any question, and therefore we'll call it a day.
That's okay. Thank you.
Okay, thank you very much. Okay, bye-bye.